I've always been interested in bone marrow transplant and how the whole process worked. The first successful transplant was done here at the University of Minnesota in 1968 by Robert Good. And even since that time, we understand very little how the transplant process actually functions. We know we infuse stem cells, and we know that the stem cells find their way back to the bone marrow, but in between the infusion and getting the bone marrow, we have very little knowledge of what's going on. And so I was very intrigued in trying to figure that out and make the process happen more efficiently. I think after spending a lot of years attending on the inpatient service and taking care of children after bone marrow transplant, it was clear to me that that critical time period after transplant, the first 30 days in which we're waiting for cells to recover, was where we needed the most help. So the main goal of the project is to accelerate blood cell recovery after a bone marrow transplant, and we're using the funds to search for new and novel compounds that might do this. When you take a child or an adult through bone marrow transplant, after the chemotherapy is given and then the transplant is given, it often takes 2 to 4 weeks for the body to recover and regenerate white cells. That is the critical time period in which a person could die from infection. So if we can figure out ways to have those white cells recover earlier, we can protect patients from infection, and that's the real goal we're going after. We zeroed in on approximately 35 initial compounds, and now we've narrowed our hits down to 2 compounds that look like they may actually accelerate hematopoietic cell transplant. Using the grant from RMM, we will better characterize very specific small molecules that we discovered using previous RMM grant funding to be able to carry this project hopefully out of the lab and back into the clinic.